So today we will discuss the physiology and anatomy of sarcomere. What is sarcomere? Sarcomere is the main contractile unit of muscle. This is the contractile unit of muscle. Let's recap yesterday's lecture quickly. That if we have a bone and muscles are attached to this bone by which thing? Tendon. What is the difference between tendon and epineurosis? Tendon is root-like structure, having root-like structure, and epineurosis have sheet-like structure. Both are tendons, right? When we have cut this muscle and see it cross-sectionally, like for example, this is cross-sectional area of skeletal muscle. Above the skeletal muscle, we have a fibrous connective tissues layer, which is made up of dense irregular connective tissues, and this layer is called epimysium. Inside this membrane, muscles will be present, and if we feel this area, we will see muscles, right? And a lot of fascicles are present inside this cross-sectional area, right? And inside this, muscle cells are present, right? We have zoom in one fascicle from here. And we can see that there is another connective tissues layer above, and this is called perimysium, right? And a lot of fascicles present inside this fascicle also, because this is one fascicle from this area, right? And if we feel this area, we will see that original muscle here, right? And a lot of fibers are present in these fascicles, right? So now we will zoom one, and we can see that there is another tissues layer on this muscle, right? And this is called endomysium, right? But inside this endomysium, we will see the sarcolemma, and inside this, thousands and hundreds of myofibrils are present. We will take one myofibril from here. This is actually sarcolemma. And what is sarcolemma? Sarcolemma is the cell membrane of muscle cell, right? Like for example, I am taking one cell from here, because this is also known as muscle cell, right? I am taking the muscle cells here. For example, this is a single muscle cell, right? This red layer is sarcolemma. So red layer is sarcolemma, and sarcolemma is actually the cell membrane made up of phospholipids, right? Above this sarcolemma, this layer is present, which is endomysium, mycelium, right? This is endomycelium, this red is sarcolemma. If I take one cell, this is endomycelium, this black one is endomycelium, and this red one is sarcolemma, right? This sarcolemma is attached with endomycelium. We will discuss this in detail. Specific proteins are responsible for this connection, right? And this muscle cell or this sarcolemma is multi-nucleated, and these nucleuses are present sidewise, right? These are nucleuses, and these cells are multi-nucleated, having a lot of nucleuses, right? And a lot of mitochondria is also present, because actin and myosin activity is dependent upon the ATPase or this ATPs, which are produced by this mitochondria, right? This is single cell which is multinucleated. Endomycelium covers this cell, and this endomycelium is made up of areolar connective tissues, right? And this sarcolemma is composed of phospholipids. So don't be confused between this sarcolemma and endomycelium, right? Now we will take one myofiber from here. These are myofibrils. If I take one myofibril, this is myofibril. Now I am going to teach you the sarcoplasm. What is actually the sarcoplasm is, right? The area between or the area inside this sarcolemma is sarcoplasm, right? So now the important point, the area between these fibers, this is actually sarcoplasm. This is actually sarcoplasm, because these long cylindrical fibers are present inside this cell, and it will take the stimulus from outside and will contract, right? And due to the contraction of these all fibers, all the muscles will get pulled on and will contract, right? So now we are going to discuss the detailed structure of this myofibril. So we will draw it a little bit larger. For example, I am taking this area. This is actually myofibril, and we are taking this like this is myofibril. This is myofibril, right? This myofibril gets striated. Myofibril having striations, having bands. You will see under microscope that there are some light and dark bands present on this, right? This myofibril. Like for example, I have this blue is showing the light band. This is light band, right? And this black is showing dark band. This concept is very important for MCQs type questions. This black is dark band, right? And on other side of this light band, another dark band is present. Another dark band is present, right? This dark band is called A band, right? And this light band is called I band. We will discuss this in detail how these bands are formed, right? And again, this is A band. A stand for anisotropic. Anisotropic. And anisotropic mean dark. I mean isotropic, and it is called light, right? One area is present in front of these bands, like this green, right? And this green area is called H zone. H zone, right? And again here the dark band is present. And here again light band. Light band, and again here A band, right? So the conclusion is that one light band is sandwiched by two dark bands, and one edge zone is also covered by two dark bands, right? Now we will divide these bands, these areas, right? So we will take two or three areas here and will divide, right? And after that we will take one sarcomere. Now we will define what is one single sarcomere, right? For example, this black is, this is A band, this is A band, and this blue is I band. This is I band, and again this is A band, right? And here is H zone. H zone, right? And here again A band and I band, right? And so on. This is A band, this is I, this is A, this is H zone, this is A band, I band, and A band, right? One line dividing this I band, like, right? And this is also, right? And one line is dissecting this area, right? This is called Z disc, and this is also called Z disc, and this is called M line, right? The distance between two consecutive Z lines or Z disc is called sarcomere. This is called sarcomere. This is the main contractile unit, right? This M line gives support to these components which made this whole sarcomere, right? In just a few moments we will know about this, that there are a lot of proteins responsible for making this red line, and this black line, right? Now these are the areas. Now we will take one sarcomere, some area of one sarcomere, and will zoom it. This Z line is responsible for attaching the thin fiber, because there are two types of fibers present in muscles. One is called thin fibers and one is called thick fibers. Thin fibers are actin and thin fibers are actin and thick are myosin. We will discuss the structure of myosin and actin also in detail, right? And the structures and elements which are important for these structure functions, right? Now this Z line is Z line because this is zigzag, like this is Z line, right? The Z line attaches the thin filaments to itself like this. These are the thin filaments which are called actin. These are actins, right? And in middle of these thick fibers are present. These red are myosin and this blue are actin, right? The actin and myosin, right? And this protein, this black one protein, it divides this area and called M line. This is called M line. Separate proteins are responsible for making this line, right? So this is the main contractile unit, because when the muscles contracted, these Z lines moves and this Z line also move, and this area covered by these actin proteins molecules, right? So now we will take one unit from here, like for example we are taking this area, right? And zoom in there, right? For understanding that how this area, how these myosin actin are interrelated to each other. So let's, for example, this is the main myosin protein. The thick filaments. Now we will just discuss this thick filaments. This thick filament is composed of several parts. For example, this is thick filament, right? And to this thick filament, here the actins are present. For example, this is Z line. This is Z line. And this is another Z line. This is another Z line, right? And 18 proteins are present These are actin proteins, right? For example, here also the actin proteins will. Actin proteins also have double helical structure. Right? And here also the actin proteins are working. These are the actin protein. Actin protein is formed of which type of proteins? Anyone? Which type of proteins they are? The G protein and F protein. They are specific. G protein and F protein. We will discuss this. This is actin, right? And double helical structures are also arising from these myosin proteins. Right? And these are surrounded by the RLC and ELC. And to these, the head of this all story is present in your Guyton book, right? On this side also we have double helical proteins. These are the myosin proteins, right? Just imagine this is the M line. This is the M line. So M line also have made up of protein. For example, these green proteins making the M line. M line is looking like this, right? And we have this black proteins present inside this myosin protein. It's present like this because myosin cannot suspend in air, right? These proteins holding this myosin on its place. Also these proteins are a very this is the one of the springy protein in body. They have, I think, 2 million molecular weight. These are long fibers, these proteins, and this is just like spring, elongated and attached to this Z protein, right? On this side also it form this springy structure and attached to this Z line, right? Now this is Z line. This thing is Z line. Let's talk about this Z line. The distance between one Z line and another Z line is Sarcomere, which is the main contractile unit of muscle. This is Z line. This is also important for MCQs, right? The distance between two consecutive Z line is sarcomere, right? This Z line, which is called zigzag line, is composed of a very special type of protein which is called alpha actinin proteins. Alpha actinin proteins. Don't be confused between actin and actinin. Right? This is another type of protein, actinin. Right? There is a need of support for this actin also. The actin is also needing support. So one protein supporting this actin also, and this protein is present in middle of these all actin molecules. Right? This black protein which gives support to this actin protein is called anyone? Nebulin. Nebulin proteins, right, which giving support to this actin molecules. One protein looping this whole actin molecule. Just take one protein from this, right? For example, here one monomer, we are taking one monomer from this. Can I remove this area? Does everyone come? ये 18 माने मतलब कोई कम. For example, I am taking one monomer from here. For example, this one. This is the single protein from actin molecule, right? Because actin molecule actually it is made like this. Actually it is two sheets of amino acids or proteins, right, like this, and one protein surrounds this actin, right? So I am taking only one from here, which is this one, and this is called G protein, globular protein, G protein. When polymerization takes place in these units, which thing is responsible for formation of these globular protein in cell? Not a ribosome. Ribosome is responsible for making these, but ribosome making these proteins for formation of actins, right? The signals are separated from their genes, growth factors, etc. Right? So when polymerization starts in this polymerization, do you know polymerization? The addition of a lot of monomers to form polymer is called polymerization. So it will polymerized, and now they are happy because they are getting friends. Right? This is now called F protein. This chain is called F proteins. F proteins, right? And why it is called F protein? Because in this form this is globular, and in this form this is filamentous. These are filaments, So they are called F proteins, and these F proteins form this whole actin protein, right? But every unit have, for example, this is the we are just zooming actin protein. Every protein have a mouth, a large mouth, right? And this mouth is responsible for holding the heads of myosins for contraction, right? But these are not opens every time. There is one thing which maintain this mouth closed. So one protein is responsible which maintain this mouth closed, and that is called tropomyosin, right? And this looping red protein you can see here, this is tropomyosin. Tropomyosin closes this mouths and not allow these mouths to hold this head of myosin, right? These protein are called tropomyosin. So all these actin molecules are surrounded by tropomyosin, right? Like that, this one is also, and this one is also, right? For removing this tropomyosin, this actin protein needs energy. It will need energy, and for that another protein binding here which is called troponin, right? So troponin is like this, right? Here this troponin is trending. If both the hands are filled, the muscle will contract, right? How? This troponin, this is troponin have three sides. Troponin have three sides. One is these feet, and these are called the actin binding site. Actin binding site mean here actins are binds. So for example, these are the troponin molecules. A lot of troponin molecules are present here. They are standing Are you people getting me? These are troponin molecules having three sides. One is actin binding site by which it binds with actin. The other is calcium binding site. This one. This is calcium binding site. Right? To this it will bind with calcium, because calcium have a key role in muscles contraction. Very crucial for muscles contraction, calcium, right? And the next is tropomyosin binding site. Tropomyosin binding site. To this site it binds with tropomyosin. On this side it will attach to troponin and actin, and on this it will bind with calcium. There are some specific names for these sites. This site is called troponin C. This is called troponin C, right? This site is called troponin T because tropomyosin attached here, and this site is called troponin A. No, it is wrong. Troponin I. Tropoi. Had you heard about tropoi during heart attacks? In heart attacks, we will test tropoi, right? When this tropoi is raised, it is the indication of heart attack. जी class was start शुइल. Right? In heart attacks, we are testing tropis. These tropis, if tropi is raised, it is indication for heart attack because this tropi is inhibitory. This is inhibitory protein. So when the muscles are not contracting, the tropi level will be raised because it inhibits the contractions of muscles, right? So that's why we test tropi in cardiac failure or myocardial infarction, heart attacks, anginas, right? So these are the site of troponin, and this is actin. This red is tropomyosin. So these are the protein. This blue protein collectively it is called alpha helical proteins. Alpha helical proteins because it forms a helical structure. But actually these are G protein and F protein, right? This is actin. Now we are going for this myosin. This is the thick filament, and this is why this is thick filament, because it is composed of 3 layers. Actin is formed of 2 layers of proteins, and this is formed of 3 layers of protein. Like for example, this is actin, the myosin molecule, right? And they have some bridges, cross bridges, which are called the heads of myosin. On other side, it also contain the bridges, right? So we are now discussing about the myosin. If we take, this is actually myosin protein, and these are the branches of myosin protein. We are taking one structure from here, right? And for example, this is the actin molecule, the myosin molecule, sorry, and this is the that helical protein. Right? Actually, these are myosin proteins. These tails are myosin proteins. Right? Here, a specific type of protein, another protein is present. I will discuss just in seconds that these are ELCs and RLCs. Right? And it attached with a this area, right? And we can divide this. This is the tail of myosin. This is the tail of myosin. This blue is the neck of myosin, and this is the head of myosin, right? The tail, neck, and head. This tail is formed of myosin protein. These are actually myosin proteins, alpha. These are also alpha, right? This is supporting protein. This is supporting protein. the neck, which is called which have two types of protein. One is regulatory light chain and the other is essential light chain protein, right? For example, this is neck. This is neck. These proteins are incorporating in this, right? On one side, for example, on one side of this, ELCs are present. ELCs. ELC mean essential light chain protein. Essential light chain protein, and essential light chain proteins giving support to this head, right? The next, present on this side, this red one, and this is called RLC. RLC protein, which means regulatory light chain protein. What is the main role of ELC and RLC? This RLC structurally support this head, and this ELC and this RLC functionally support this head because it attached the inorganic phosphate from ATP, because one side in this head, this is called ATP binding site, having ATP binding site, and this one is, this is actin binding. On actin, it will attach to this mouth. This is the myosin binding site on actin. So this is the actin binding site, right? When this will always in this form, enclosed form, but when ATP came here, it activated. Like for example, it will become like this, right? It will open when it takes one ATP and converts it into ADP. It will take energy and this head will become open, and now it can bind with the mouth of that actin protein and will contract. When these both are joined, it starts contraction, right? We will discuss this contraction mechanism in after neuromuscular junction, right? But this is just in structural overview, right? So this is the structure of myosin, right? The next thing, the spring protein. This is very important. This spring protein is very, very, very important because when the muscles contract, for example, this protein will go in this direction. It will contract, right? These springs will also contract, right? And when these springs are contract, after inactivation of these heads, after inactivation of this contraction, these springs are responsible for giving the position to these myosin, all position, right? So these are also known as springy protein, but the name of this protein is called titin. These proteins are called titin proteins. Titin proteins. Titin proteins are the proteins which attach the myosin with Z line, right? Next, this M line. M line is made up of two functional protein, two structural protein and one functional protein, right? What are these proteins? Any one of you? Have you heard about these proteins, or only just the M line? Just M line. The protein which are responsible for making the M line is myomesin. The first one is myomesin protein. The second one is C proteins. C proteins. And the next one is creatine kinase. These are enzymes protein, right? So this one is functional protein, functional, and these two are structural protein, right? And the role of this creatine kinase is in metabolism, right? Because skeletal muscles are responsible for metabolism also, because its exercising or the daily work is dependent upon these muscles. It need a lot of metabolism, so these creatine kinase and other proteins present in skeletal muscles are responsible for this. uh, Creatine kinase are responsible for that metabolism, and this myosin and C protein is responsible for making this M line. And what is the role of M line? M proteins, it holds the myosin on its maintain position, right? If it will break, it will affect your muscle, right? The contraction of your muscle, right? We are just uh, discuss that in just a few minutes, right? And I have told you here in sarcomere that this sarcolemma is attached to endomysium, right? So now, having a look for that, what is the connection between these actin molecules, the sarcoplasm, the sarcolemma, to the endomysium, right? Dauran come the side. ده هو برونيك اشتاك انا ده حرصه Now let's see. For example, this is the myosin protein, right? This green is M line, M proteins, right? This black is titin proteins. Right? This red is Z line. This is Z line, right? And these blue are actin molecules, right? These black are nebulins. These are nebulins, right? And these are the tails, of myosin, right? And these are the heads of myosin. We will discuss the function of these heads in muscles contraction in detail, right? No, we are not wasting the time on this, right? Here the endomysium is present, endomysium, right? And here the sarcolemma is present, right? Like this. This black is endomysium, this red is sarcolemma, and this is the internal structure of this cell, right? Some specific proteins, this is the interstitial fluids. Do you know these are the extracellular environment. Some important proteins have connection between this endomysium. For example, here is a bunch of protein present. Like for example here, right? And these proteins are attached to this bunch of protein. What is the name of this protein? This is called dystrophin proteins. Dystrophin protein. And these proteins are responsible for dystrophy of muscles, right? How? For example, if this connection is damaged. Mutation takes place. Two types of dystrophies are here. For example, these proteins are responsible for muscle dystrophy. Do you know muscle dystrophy? Muscle dystrophy means the hardness of muscles. When the muscles become fibrous, right? When the muscles become hard and they cannot work, then physiotherapy is required for that muscles. Then, right? So this muscle have two types. One is called, I think, Duchenne dystrophy, muscle dystrophy, and the second one is called brick muscles dystrophy. Right? This is called nonsense. This is called missense. There are two things. When something is present and not working, this is one condition. And the second condition, when that thing is not present. Right? For example, if we have insulin in our body and they are not working, this is called diabetes mellitus 1. And when the insulin are not present, right, this is another type of diabetes mellitus. Like that, here in Duchenne MD, this is abundant. This is mean more severe, right, and occurs in the age of what is in your mind? Dystrophy is in the age of 40 or 50 years. 40 years. Muscle hardness. This caused in 5 to 6 years. In 5 to 6 years age, the muscles get dystrophies. And this is more severe because this is also known as nonsense muscle dystrophy. And in this condition, the genes responsible for making dystrophin proteins, right, mean dystrophin genes are not working and they are not signaling to form dystrophin proteins. The other, what is called break, this is less severe. It is bad. Even it is bad, but this is less severe and caused an age of 10 to 20 years. Right? When you are playing, you are a teenager. Right? In this age, you can caused by this break muscle dystrophy. In this case, this is called missense muscles dystrophy, because in this the proteins are highly coiled. Because proteins are in coiled form or in having quaternary structures, when these proteins are more tight, it will change their functions, right? So like that, this protein is in modified form. Then, for example, if I have this protein like this, and when it tightened, it become like this, it modified, and the function of this protein will also affect. In this case, this is called the break muscles dystrophy. Right? These are the clinical points related to the muscles. Right? Muscles dystrophies. This is one. This dystrophin protein is also responsible for cardiac myopathies. Cardiac myopathies mean the muscles dysfunctions in heart. Right? When the muscles are not working in heart, not contracting, and we cannot give physiotherapy to heart. Can we? No. It will affect your heart very badly. Right? And this dystrophin proteins are responsible for myopathy of heart muscles also. Right? So this is the whole story of sarcomere. After this, we will discuss the nerves. For example, nerves is coming from spinal cord, the ventral horn, right? And it will innervate this cell. For example, this is our cell. This junction, this is neuron and this is muscle. So this junction is called neuromuscular junction. Right? We will discuss this neuromuscular junction in detail, because the general overview of muscle contraction is like that. When it gets stimulus from brain, it will release acetylcholine, and acetylcholine there are a lot of extrinsic protein channels which are activating on acetylcholine. Right? And inside this cell is rich in potassium, magnesium, right, chloride, calciums, right, and outside sodium, a lot of sodium is present outside the cell, right. So when acetylcholine comes here, it will activate a lot of channels and sodium will rush into the cell and the cell become depolarized. And when the cell depolarized, there are a lot of biological changes will take place in that muscle cell. We will discuss that in detail after neuromuscular junction. So the coming topic is neuromuscular junction. Doa makawai. This is Abde.